Hi everyone, my name is Yurui, and today I'm presenting my research project on the discovery of TBL1XR1 drug-like molecules for the development of inhibitors or degraders. This project was conducted under the supervision of Dr. Shapira at University of Toronto and using the molecular software ICM Pro by MoSoft. Transducing beta-like 1X-linked receptor 1, TBL1XR1, also known as TBLR1, is a well-known core component of the NCOR smart co-repressor complexes. When it binds to the unliganded nuclear receptors, it is responsible for transcriptional repression. In addition, one study has proposed that TBL1XR1 functions as an E3 ligase when binds to liganded nuclear receptors, recruiting the E2 enzyme UBCH5 for the ubiquitin-mediated proteasomal degradation of the co-repressor complexes to exchange for co-activators, which will promote transcriptional activation. TBL1XR1 was reported to be overexpressed in multiple solid tumor types, including colon cancer, prostate cancer, breast cancer, hepatocellular carcinoma, and pancreatic adenocarcinoma, and was correlated with tumorigenesis, tumor progression, and development of chemoresistance, which made it a potential target for anti-cancer agent development. For example, it has a role in cisplatin resistance of nasopharyngeal carcinoma and esophageal squamous cell carcinoma by activating the NF-kappa B pathway. In addition, it has been found to be overexpressed in lung squamous cell carcinoma, colon cancer, and breast cancers, promoting tumor cell proliferation and tumorigenicity by activating Wnt beta-catenin signaling pathway. In previous studies, siRNA knockdown of TBL1XR1 in colorectal cancer cell lines inhibits Wnt beta-catenin transcription by blocking beta-catenin binding to T cell factor 4 in the promoters of target genes, and thus inhibiting cancer proliferation. Similarly, in another study, the shRNA knockdown of TBL1XR1 in pancreatic adenocarcinoma cell lines and mouse models significantly downregulated PI3K and AKT protein expression and induced the pancreatic adenocarcinoma cell cycle arrest at G0-G1 phase by regulating cell cycle related genes, for example CSC25A. These data suggest that inhibiting TBL1XR1 might be a potential anti-cancer strategy for treating solid tumors. Aside from 4 ongoing clinical trials, there is no information available about small molecule inhibitors for TBL1XR1. This lack of information indicates the scarcity of small molecule inhibitor development for TBL1XR1 and highlights the importance of this project. Alternatively, taking advantage of TBL1XR1 as an E3 ligase, targeting protein degradation with a small molecule drug can also aid anti-cancer treatment. Proteolysis targeting chimeras, PROTAC, are bifunctional hybrid compounds that bind to both E3 ubiquitin ligase and the protein of interest, abbreviated as POI. It achieves this ternary complex through 2 recruiting ligands connected by a linker. In this case, one ligand will be specific for the protein of interest, and the other drug-like small molecule will be specific for TBL1XR1. Upon binding, PROTAC will facilitate degradation of the protein of interest through E3 ligase mediated recruitment of E2 enzyme and subsequently ubiquitin proteasomal complex, and thus selectively knock down the protein of interest level. Compared with conventional occupancy-driven inhibition by small molecule inhibitors, PROTACs promote non-native target degradation in a catalytically driven mechanism of action, meaning that one PROTAC molecule can drive multiple rounds of target degradation. Therefore, a decreased PROTAC concentration is sufficient to generate therapeutic effects and subsequently reduce the possible undesirable off-target effects. Furthermore, one study has indicated that TBL1XR1 works in the same pathway with beta-catenin in regulating transcription activation of cell proliferation genes, suggesting the possibility of targeting beta-catenin degradation through TBL1XR1 based PROTACs. Overall, these data indicate a potential for generating TBL1XR1 based PROTAC for target protein degradation in anti-cancer therapies. The human TBL1XR1 gene is located on the long arm of chromosome 3 at position 3q26.32. Its mRNA is ubiquitously expressed in many human tissues, with higher levels of expression in thyroid, prostate, and breast tissues. TBL1XR1 protein contains at least one homology domain and an F-box-like domain at the N terminus, and 8-bladed WD40 repeat WDR domain at the C terminus. The least H domain is important for oligomerization and histone binding, which is essential for the stabilization of NCOR smart co-repressor complexes and is required for efficient transcriptional repression. The F-box-like domain is essential for the recruitment of the E2 enzyme UBCH5 for the degradation of NCOR smart co-repressor complexes, allowing the exchange for coactivators and subsequent transcriptional activation. 8-bladed beta propeller WDR domain mediates protein-protein interactions and was found essential for interaction with substrate proteins that get ubiquitinated and degraded, which suggests a likely binding site for drug-like molecules. The structure of the TBL1XR1 family is evolutionarily conserved with high degree of similarity across species, suggesting similar molecular functions. Taking into account the background information, we aim to apply the structure-based and ligand-based virtual screen that use the information on a peptide-bound structure of TBL1XR1 protein to select a putative drug-like ligand candidate for the generation of TBL1XR1 based PROTAC or small molecule inhibitors. Virtual screening is a technique for selecting hits from large chemical libraries through computational means based on knowledge about the protein target, which refers to structure-based virtual screen, SBVS. Usually use the information on the 3D structure of the target receptor, or known bioactive ligand, which refers to the ligand-based virtual screen, LBVS. In structure-based virtual screen, molecular docking is a key tool used to screen compound libraries based on chemical complementarity between ligand and target protein to determine the predominant binding poses between ligands and the target receptor. It uses a scoring system that provides a docking score to assess how favorable the pose is. Usually, the lower the docking score, the better the binding pose. Alternatively, in ligand-based virtual screening, pharmacophore modeling is utilized, which relies on the pharmacophore features of the chemical template derived from a known ligand of the target receptor. A pharmacophore is defined to be an ensemble of steric and electronic features that is necessary to ensure the optimal interactions with a specific biological target. The pharmacophore of the compound can be studied by the atomic property field (APF) method developed by Mosoft. It models the physiochemical properties of the compounds, and the APF models for TBL1XR1's endogenous ligand is shown in the match view here, where the blue represent hydrogen-bound donor, red represent hydrogen-bound acceptor, yellow represents lipophilic portion, and the white represent large size of the molecules. And in this case, the rapid isosterol discovery engine, the Receptor, is used for the ligand-based virtual screen. The library is screened based on compound similarities to the template pharmacophore. In this case, the higher the right score, the more similar the pharmacophore property is to the template. Virtual screens were chosen over experimental screens due to their time and cost effectiveness. It is crucial in the selection of hit candidates when the access to routine experimental screens is unavailable. Also, the compound library for experimental screen is usually 1,000 times smaller than the virtual screens due to the funding restrictions. In addition, unfavorable compounds can be eliminated at an early stage in drug discovery for the virtual screen method, and the top 20 to 30 hit candidates will be useful in vitro testing, thereby reducing cost and saving times compared with the experimental screen. Here is the overview for the whole project. Since the large-scale compound library contains 19 billion compounds, which will take days for screening, we first identify key interactions through small-scale compound library analysis and optimize the most promising chemical template for large-scale in silico analysis. The chemical template is then used for large-scale ligand-based virtual screen, and then the output from the large-scale ligand-based virtual screen or the Receptor will be used as the input for the structure-based virtual screen, so that we can bring together information from structure-based virtual screen, which assesses how favorable the binding poses of the hit compounds are to the target receptor, and from ligand-based virtual screen, which assesses how similar the pharmacophore properties of the hit compounds are to the chemical template. After that, 3 representative hits are selected based mainly on the docking score and the right score, with the RTCN score as supplement. So for the hit 1, it will selected to have the lowest docking score, hit 2 selected to have the highest right score, and hit 3 have the best combination of both scores. As mentioned before, the WDR domain of TBL1XR1 mediates protein-protein interactions and is a suitable pocket for small molecule binding, which is our main focus. The top interaction surface is evolutionarily conserved in the WDR domain across species and is highly electronegative, enabling recognition of a cluster of basic residues. Since there's no available crystal structure for the human TBL1XR1 WDR domain and its known ligand interaction complex, the crystal structure of the revised mouse TBL1XR1 WDR domain in complex with its endogenous ligand MECP2 was obtained from the protein data bank instead. The structure of mouse TBL1XR1 was solved by molecular replacement using coordinates from human TBL1XR1 as a search model, and the revised mouse TBL1XR1 WDR domain achieved 99.5% identity to human TBL1XR1 WDR domain. And here's the superimposed model for them. Before we started the virtual screening processes, we did an interaction analysis between the TBL1XR1 WDR domain and its endogenous ligand MECP2, and we found proline 02 indicated in the magenta color, lysine 04, lysine 05, and arginine 06 form the core of interaction. Arginine 06 forms the salt bridge with glutamic acid 171, indicated in the green dots, and proline 02 is surrounded by tyrosine 395, phenylalanine 420, and tyrosine 446, corresponding to the hydrophobic interaction core. Two basic residues, lysine 04 and lysine 05, are essential for hydrogen bond formation. Lysine 04 forms the hydrogen bond with the backbone carbon of asparagine 353, while lysine 05 forms a hydrogen bond with the amide side chain of asparagine 353. In addition, lysine 05 is further stabilized by forming a hydrogen bond with the side chain of aspartic acid 369, which will be our focus for later in silico analysis. After small-scale structure-based virtual screening, 347 compounds were returned with docking scores ranging from -10 to -23.76. The hotspots for hydrogen binding were identified by selecting the most prevalent residues involved in hydrogen bond formation. Based on this criteria, the phenyl OH group of tyrosine 395 and the backbone Ne group of leucine 272 are the major interaction hotspots in hydrogen bonding. Similarly, hydrophobic interaction hotspots were identified by selecting the most prevalent residues with interaction distances lower than or equal to 4 angstrom. In accordance with this criterion, isoleucine 174, tyrosine 395, and phenylalanine 270 are the major interaction hotspots in hydrophobic interactions. These informations all contribute to the later chemical template design. Based on previous literature reports and the small-scale in silico analysis of TBL1XR1 MECP2 complex, proline 02, lysine 04, lysine 05, and arginine 06 are found to be 4 key essential residues contributing to most hydrogen bond formation and hydrophobic interaction. In order to design the chemical template for large-scale in silico analysis, several combinations of residues were tested in the small-scale research, and the template returned the most hit compounds would be selected for further occupancy value editing. The chemical template was designed to reduce the dissolution penalty, and thus lysine 05 and arginine 06, which form hydrogen bonding near the receptor surface of TBL1XR1 and have greater potential to form hydrogen bonds with water molecules, are excluded. Finally, a chemical template derived from proline 02, isoleucine 03, and lysine 04, which produce the most hit compound, were selected for occupancy editing. Proline 02 and isoleucine 03 are the main residues for hydrophobic interactions, and on the left you can see that these two residues are lipophilic, indicating by the yellow color, corresponding to the driving force of binding, while lysine 04 contributes to hydrogen bond formation, corresponding to the binding specificity. And here you can see the pharmacophore property here is blue, so indicating it's a hydrogen bond donor. Occupancy value of essential side chains were set to 3 to set a preference for large-scale research. Since the compounds resulting from large-scale research and structure-based virtual screen do not have a specific distribution pattern, hit compounds with the lowest docking score or the highest right score or the best combination of the two are selected as representatives. In common with the three representatives, they all form hydrogen bonds with the backbone carbonyl of asparagine 353, which is consistent with previously reported literatures. And all the hydrogen bond acceptor donor pairs with a distance lower than 3 angstrom, which falls within the acceptable range from 2.7 to 3.3 angstrom, could potentially increase the binding specificity. Also, they form multiple hydrophobic interactions with residues that match the literature review and the small-scale in silico interaction analysis, including phenylalanine 173, tyrosine 395, threonine 229, and tyrosine 446. And space filling models do not show any intermolecular clashes. In addition, all three representatives have cyclic structures introduced to the side chain of the lysine 04 region. The addition of cyclic structures increased the hydrophobicity and rigidity of the compounds, which enhanced the binding force and reduced the entropy penalty, and subsequently increased the potency. However, the increased rigidity makes the side chain less flexible, which could make the hit compound hard to fit into the receptor pocket. And thus, further in vitro tests are needed to determine their true potencies. In the case of each representative hit compound, hit number 1 forms a new face-to-face pi-pi stacking interaction with phenylalanine 173, which is the most unstable type of pi stacking interactions due to the electron cloud repulsion. But overall, this introduction of face-to-face pi stacking interaction enhances the driving force of binding as well as specific recognition of binding. Similarly, hit number 2 also form new pi-pi stacking interactions with two different residues of TBL1XR1. It forms a parallel displaced pi-pi stacking interaction with tyrosine 395 and a T-shaped pi-pi stacking interaction with phenylalanine 487. The parallel displaced configuration and the T-shaped configuration have the strongest quadruple interaction among all types of pi stacking interactions, as the parallel displaced configuration reduces the repulsive interaction by displacing the negative electron clouds, and the T-shaped configuration has positive sigma bound facing negative electron clouds. In addition, there is a fluorine atom attached to the phenyl group of hit number 2 The addition of halogen atoms represents the potential for further structural activity relationship analysis. Hit number 3 does not have large modifications and aligns the best with the chemical template. The hit has 2 fewer hydrophobic interactions, namely phenylalanine 270 and phenylalanine 487, compared with hit number 1 and hit number 2. However, it has the lowest RTCCN score among the 3 representative hits. RTCN score is a deep learning-based model generated by Mosoft. The score represents how good the predicted protein-ligand interaction is, where the lower scores are better. Generally speaking, hit number 3 with the lowest RTCN score, essential hydrophobic interactions, and key hydrogen bond still has the potential for later SAR editing. And here is a notification. Although ICM User's Guide recommends plotting the docking score and RTCN score together to prioritize the hit compounds, we found an overlap between the combination of docking and RTCN scores and the combination of docking and right scores. As a result, we remain committed to prioritizing the hit compounds based on their right score, docking score, and both for further SAR editing and in vitro experiments. Generally speaking, the top 3 hits with various types of new interactions, such as pi-pi stacking interactions, serve as the starting point in searching for small molecule inhibitors or PROTAC for TBL1XR1 protein. The next step is to identify the most populated scaffolds shared by most hit compounds and apply Mosoft ligand editor for hit optimization. Also, RTCN scores can be used to assess how good the modification to the hit compounds. Then, the top 20 to 30 hit compounds will be purchased for in vitro efficacy and potency tests. Future perspective. In this project, one methyl group of isoleucine 03 is missing from the crystal structure and thus could affect the hit selection process. We may reference the predicted structure from AlphaFold for improvement. In addition, the overexpression of the TBL1XR1 protein was correlated with many types of solid tumors. There are currently no small molecule inhibitors available, and thus this project is a good start due to the scarcity of inhibitors. However, potential problems like loss-of-function mutations of TBL1XR1 are found in mental retardation and B-cell lymphoma. Affected residues include tyrosine 395, phenylalanine 420, and tyrosine 446. Based on previous report and in silico analysis of this project, tyrosine 395 and tyrosine 446 are the 2 most prevalent residues involved in hydrophobic interactions, demonstrating the relevance of these 2 residues. Therefore, while no specific mutations of TBL1XR1 have been reported in solid tumors, mutations of these 2 residues are of great attention in developing small molecule inhibitors. Moreover, as described before, the ubiquitous expression of TBL1XR1 mRNA in multiple tissues, suggesting a requirement for the targeted delivery system. Furthermore, TBL1XR1 is known as the key component of the co-repressor complex regulating transcription repression when binding to the unliganded nuclear receptors. However, the PROTAC design emphasizes its structure as an E3 ligase, mainly responsible for transcription activation. Further studies are needed to reveal the mechanisms directing TBL1XR1 toward its E3 ligase function. In addition, although previous studies have indicated that TBL1XR1 has the E3 ligase role for E2 enzyme recruitment and subsequent degradation of co-repressor complex, this finding was not reproduced by later studies. More studies are required to reproduce the finding of TBL1XR1 as an E3 ligase to further support this proposal. In this case, the PROTAC design is still a long way from completion. And this is the end of my presentation. Thanks for listening.